we don't forget. So, a little bit of the history of something, and then why it's important, some of the methods that they used, the results, and then the mechanism. So we'll work through like Renin angiotensin system today, and what we'll do is we'll just kind of start off with a little bit of the history of it, and then we'll dissect a kidney. Hopefully that's going to go okay. We'll take one of the sheep's kidneys that we did in one of our earlier labs. We'll identify all the constituent parts on the exterior of the kidney, and then we'll cut it open. We'll do a coronal cut. We'll look at the pyramids, the renal columns, papillae, the calyx, the minor major, the renal arteries, veins, and the ureter and the renal pelvis. And then we'll look at, from there, we'll look at the adrenal that lives on top of the upper pole of the kidney. And then we'll talk about how originally somebody ground that up and put it on things and got a pressor reflex, which means an increase in blood pressure. So we'll work all the way through that and then get it to the aldosterone mechanism. And hopefully it'll all come together. But what I'm going to end up doing is sharing I've used my phone as a document cam, so hopefully it's going to work out okay. And from there, we'll be able to do the dissection and then hopefully do like we would do in class, where you would take notes. All right, is everybody on board? We good? All right. So if we're good, let me do the first thing first. All right. So that's not the one I wanted. Let me share something with you really quick. Okay, over here, this is an example of a works cited that I created just for our example for today. It had some of the papers that I looked at to look at the discovery of the Renin angiotensin system, and then some random papers there to give you an idea what a works cited is going to look like. And I just, using the citation manager, I copied and pasted the correct citations from the paper, from the website that I got from. Either or is just fine. When you've only got 4 or 5 different sources, cutting and pasting is going to work okay. But when you get to the level of a master's degree or dissertation or a senior final project, a citation manager is going to really help you out. Okay, so we've got that. This is what a works cited should look like. I also wanted to show you case study. I posted one an example of our case study with the spinal cord to D2L this morning, and it looks like this. One second. So this is the format I want you to take with it. Give me the situation that happened and some questions, and then the correct answers. So think of some interesting disorder and come up with 5 to 10 thoughtful questions that are correct, and that'll be worth 5 points extra credit potentially. Okay? Any questions on, let me see. Any questions on the case study? So yeah, your situation's not as long, that's fine. As long as you describe the situation and let us know what's going on, let your audience know what's going on so that they can answer the questions from there. We'll be just fine. But the point is, I don't want to do a lot of research to make sure your point case study is up to snuff versus the amount of research I'll do to make sure your 130-point final exam is correct. Does that make sense? So it's a, uh-oh, I'm getting some power fluctuations here. we could be in for trouble. So does that make sense? The amount of effort that you should spend on things should be at this point like 98% of your effort on your final exam paper and maybe 2% of your effort on this. Okay? Does that make sense? If you were to look at how to spend your time effectively. All right, any question on the case study? Any questions on a works cited? No? You ready to venture into the unknown? So I'm going to try to do this in a way just for technology. So if things go really south, you'll understand what I'm trying to get to and be patient with me. So I'm going to I'm going to keep the microphone on in here so you can hear me. I may turn it off depending on what happens with the app because I'm sharing a different app over here. So if I start sharing this, you may hear, do you hear me twice? So if somebody could just let me know. Okay, so you only hear my voice once? Okay, good. And you can see my hand? All right, well, let's do something else here. Okay, so I'm going to turn this part off, the webcam, because you don't want to see my bald spot while I'm dissecting a kidney. So we're all good with that? You can see that? Thumbs up, thumbs down? All right. So rubber gloves. We don't want to do without that. Cool. How about that? I didn't expect it to work. So let me get my cup of coffee, get a little drink before we get started, before we get into science and my man cave. Okay. So let's talk about today we're going to talk about the renin angiotensin system. This is a mechanism that I find fascinating. It takes into account all of our different systems to control blood pressure. Where we bring in the lungs, we bring in the liver, we bring in the bloodstream, we bring in the kidneys, the adrenal gland. We end up tickling the hypothalamus at the end of it to stimulate some more hormones. But at the end, it's a really, really neat mechanism to bring our blood pressure back up. And this mechanism was teased out at the beginnings of it by a guy named Tigerstead and Bergman back in 1898. They were looking at kidneys and they were trying to find ways of, figuring out what was in it. Back in the 1800s, back then, there was a lot of science going on where they would take an organ and grind it up, put it into solution, and then put that solution on top of other things and see what would happen. So do you remember our lab when we looked at thyroid releasing hormone, stimulating hormone, adrenal cortical releasing and stimulating hormones, and we looked at the feedback loops between that? That was essentially what they were doing. So they were putting pieces of organs or endocrine organs on other things and seeing what would happen. So Tigerstead was friends with Alfred Nobel, and he was put up, I believe he was nominated twice for his work. But what ended up happening, making it really interesting, was that they set the stage for it, and we get, the adrenal gland at the top of it. And when they found out that was giving pressure effects, it set the stage for this guy named Goldblatt back in 1934. Goldblatt ended up using a canine model. He used dogs and he would clamp the renal artery, one of these arteries in here. And by clamping the renal artery, it reduces blood flow into the kidney. The kidney sensed that as a reduction in blood pressure, and it compensated by producing a chemical called renin. And renin would be released into the bloodstream and bind with another chemical called angiotensinogen. So they started to tease this mechanism out because they were able to clamp this renal artery and prevent blood getting into the dog's kidneys, which was then sensed as a lower blood pressure. So that made it kind of interesting. We got it going there. So when we look at kidneys, though, we need to look at it from the aspect of, which is it? Is it a right kidney? Is it a left kidney? Whose kidney is it? What are the constituent parts of this? So we notice that we have, this one's a lot easier to see compared to the one I was working with yesterday. Had a lot of fat in it. on the one yesterday. This one is a little bit easier to see. So we want to talk about a few things on the kidney from the outside. The kidney lives between our thoracic 11 vertebrae and our L3 vertebra. So as it sits in there, we have a right and a left one. The right and left have a little bit different function. The left is going to have on the vein, on the renal vein, it's going to have a drain for the gonad vein. I can't remember the name of it off the top of my head. It's the gonadal vein. It drains into it as well on the left side. But what we see is we have to determine what's up and what's down. So the order, what's anterior, what's posterior. So if we look at it from the aspect of anterior and posterior, we're going to have to identify what the renal vein is, where the renal artery is, and what the renal pelvis looks like. So the renal artery is always in the middle, and the renal pelvis is this area right in here, which ends up coming into the urethra right there where urine is collected. So this big gap right here, this is the renal vein inside here. So this sets us up here, letting us know that this side here is the anterior. This is the posterior side on that, because the renal vein, renal artery, and then renal pelvis. And then we can figure out a little there, because the ureter points down. We are looking at a right kidney. So this is a right kidney at this point. So the entrance of the renal vein, the renal artery, and the renal pelvis, or the ureter right here, is entering in through this hole right here called the hilum, right in here. Okay, so we have two poles on a kidney, the upper, and then we have the lower. We have an anterior, and we have a posterior side. We have two borders. We have a medial border and a lateral border. And on top, we have an adrenal gland right up here. So this is the external anatomy of a kidney. But in order to see the internal side of it, we need to take a look at it, and we can cut it open pretty easily. We do this with what we call a coronal cut, the corona. And remember that the kidney's surrounded by a tough capsule. And then we cut, take our scalpel, cut around. There is a little bit of resistance. Hopefully I don't cut my fingers off. As you notice, I wasn't playing guitar all that well this morning. I haven't practiced in quite some time. So finger dexterity isn't quite there. All right, so we're cutting this up like a piece of steak almost. But we want to take care to not get in and destroy the hilum. So we're going to spread it with our fingers a little bit. And there we go. And then we can cut. And we can see that the renal sinus, this area right in here, this hole, is mainly contained and filled with fat, as well as the entrances and exits for the renal vein, renal artery, and urethra. Okay, so now that we're inside the kidney, we get to see what things look like a little bit. So there's some main functions in here. We got our ureter right there coming out. Urine's going to collect and drain. We have a renal pelvis. And this in here is our major calyx. Our minor calyxes come in here and form ducts. And we have our externally here, we have our cortex. You can see the cortex surrounding it. And we get a little bit darker shade here. That's the medulla. And if we look really close, we can see the renal pyramids right here. And the renal pyramids are filled with renal papillae. And between the renal pyramids are renal columns. And then we can see right here one of the minor calyxes right there, this white part coming in there. So these calyxes all drain. I think they drain from like 13 and they enter in, so they can collect urine and drain it into your ureter. These are the main parts of the kidney right in here. Bring it up to you a little bit. You can see a little bit better. If we took all this fat out, you'd start seeing a little bit more of the entrances, the arteries, and the veins in your ureter. Maybe we can get in there and get into the tube and actually go through it. There we go. So you can see the tube there, and such. And then if we make one more cut, just to see what a cross section might look like. Just tear that renal vein off. All right, and this is a cross section. Can't really determine right in here. These are our the renal pyramids. This is the cortex. This is the medulla right inside here. All right. So that's our basic dissection of a kidney. Everybody can see that okay? Is that all right? So this part right up here, this is our adrenal gland, right up here. So Tigerstedt was grinding this part up here and then putting it into a solution and then putting it on other organs and seeing what happened. Okay. Any questions on the kidney? Anything else you want me to do to it before I put it away and move on? I will say I'm getting a little lightheaded from the fumes. All right, going once, twice, 3 times. All right, we're going back into the bag. Oh, goodness. Oops. Smells terrible. You guys remember that smell, don't you? All right. Okay. So is that okay? Yes, I'm. If you really, Elena, if you get into it, you would really see that there is some. If we put it under a microscope and put the tissue under that, you would definitely see a difference in the adrenal glands. And it is so much easier for the book to draw them separately, because if they drew it as just it looks like the same tissue, nobody would believe you. That's kind of my thought on it. Okay, so we've run ourselves through the history a little bit of the kidney. And what I did want to run through real fast is that there is a pathway that things take. We have blood that enters into the kidney, into the hilum via the renal artery, and it leaves via the renal vein. But in the meantime, there is a part of the kidney called the nephron. And the blood flow, the capillaries surround the nephron. And the path of urine, once the blood has been filtered, it goes through the papillary duct in the renal pyramid, through the minor calyx, to the major calyx, to the renal pelvis, to the ureter, and then out the urinary bladder. That's just the macro pathway of it, since we are looking at the actual gross anatomy of a kidney. So Tigerstedt's figured that out. 1934, Goldblatt figured out a little bit more to the situation by clamping the dog's kidney, the renal artery. So blood was not able to get in. It wasn't being filtered. The kidneys, the juxtaglomerular apparatus, sensed that as a decrease in blood pressure, produced renin from the kidney, which got into the bloodstream, bound up with angiotensin 2 from the liver, and started the process of long-term control of blood pressure, which is really the theme of today. So let me grab a piece of paper so we can get started with this. All right. Golly, It looks just like class, but smells like the lab, I have to say. So the story of increasing your blood pressure. It's a pretty interesting story because we have to rely on many different control factors of blood pressure. It's a negative feedback loop, and the kidneys are one of the main lines of defense when we have trouble with blood pressure. So the kidney is going to sense a decrease in blood pressure or a decrease in blood volume. This is interesting to note because a decrease in blood volume or a decrease in blood pressure are really the same thing. So if you have a hose and you have your finger at the end of it, and you are using your finger to increase the pressure inside the hose, you can do it that way, or you can increase the volume of water in that hose to increase the pressure. So the kidneys are really sensing any changes in blood pressure. So for example, if you got your leg cut off, the kidneys are going to sense that loss of blood as a decrease in blood pressure, and they're going to fire up the renin angiotensin system in order to combat your low blood pressure. Because at the end of this system, at the end of the RAS system, is to produce angiotensin 2. That's the goal. But we got to do it through, just like everything else in our body, we don't do it in our direct way for the most part. We go to a very roundabout way so that we have a little bit more control over it. Because angiotensin 2 is a very powerful hormone. It's a peptide hormone that's dissolved in plasma, which means it can travel throughout your bloodstream. It targets the adrenal cortex to produce aldosterone. It targets the arterioles to vasoconstrict them, to make the pressure. Remember, if we change the total peripheral resistance by constricting them, we're going to increase our blood pressure. It's going to end up in the medulla oblongata, and it's going to stimulate our blood pressure reflexes. It's going to let us know our baroreceptors are working or not. And then lastly, it's going to end up in the hypothalamus and tell us we're thirsty and increase our vasopressin. So this is a lot of things this one little chemical ends up doing. So the pathway is, let's start up here. We sense a decrease in BP or blood volume. And we sense this in two ways, the kidney and baroreceptors. So we've got two methods right there. And the baroreceptors, if they sense any decrease in pressure, they're going to increase sympathetic activity, and this is going to trigger our heart rate to increase, all the things that are associated with sympathetic activity in order to increase our blood pressure. It's also going to interact with the granular cells of the kidney, and then we'll produce something very similar here in a second. So in the kidney, this decrease in blood pressure is sensed as a decrease in glomerular flow rate. And when we sense a decrease in glomerular flow rate, this just means that there is a decrease in blood flow into the glomerulus. So there's less blood being filtered. This kidney senses this and says, uh-oh, this ain't good. We need more blood. We need to keep the filtering going. So what we end up doing then is we're like, okay, we got to increase this. So we're going to decrease salt concentration transport across the macula densa. Okay, and then we're going to tickle the same granular cells that we were over here. And these guys are all going to make, the granular cells are going to make renin. This is the first ingredient in our blood pressure control. We have renin. Okay, so renin now is being produced because we've sensed that there is some kind of alteration in blood pressure. Something went haywire. So the liver over here, the liver, it's over there always, it's happy. It's just filtering things, getting the toxins out of it, making everybody feel a lot better. And it constantly is producing a chemical called angiotensinogen. And this chemical ends up angiotensinogen ends up getting into our bloodstream. So this is our blood right here. This enters in, so angio. And then renin over here. And then we get renin. So since angiotensinogen is always floating around at a certain concentration, the liver is always making it. And the chance that our BP has decreased or our blood volumes decreased, we go through this chain of events to produce renin. Renin then reacts with angiotensinogen because renin is an enzyme. Angiotensinogen binds up with renin, and they undergoes a change to angiotensin 1. So remember, we need to get to angiotensin 2, which is our miracle drug. Does a whole bunch of things. So angiotensinogen plus renin have now converted to angiotensin 1. So remember, our bloodstream is going to flow through our whole body. Our cardiac output is going to tell us that 5 liters of blood are circulating throughout our body at any given time. But remember, we need to get CO2 out of our blood and oxygen into our blood, and this happens at the lungs. So our lungs over here, lungs are right here, our angiotensin 1 comes into there, and it encounters another enzyme. We have an enzyme called ACE, angiotensin converting enzyme. So if you have hypertension, there's drugs called ACE inhibitors. ACE inhibitors inhibit ACE from converting angiotensin 1 to angiotensin 2. This is what ACE does, an angiotensin converting enzyme. So in the case of hypertension, instead of angiotensin 1 coming into an angiotensin converting enzyme and being converted from angiotensin 1 to angiotensin 2, the drug comes in here and mimics angiotensin 1 and prevents angiotensin 1 from binding an ACE. So we have angiotensin 1 floating through our system that's not been converted to angiotensin 2. So we don't get the effect of increasing our blood pressure from angiotensin 2. So in this case, the person already has high blood pressure. We want to prevent it from getting higher. Angiotensin 2 is the end result or outcome to increase our blood pressure. So if we inhibit the conversion of angiotensin 1 to angiotensin 2, we've now prevented blood pressure from increasing, and we can keep it a little bit more stable. So at this point, this enzyme that lives in our lungs, ACE, has reacted to the angiotensin coming 1, converted it to angiotensin 2. And now angiotensin 2 is into the bloodstream. Angiotensin 2, I'm going to draw a second little arrow here and what it does. So now angiotensin 2 is now in circulation, and it has a number of effects. We can vasoconstrict, which is a result of increased sympathetic. We have increased aldosterone. We can increase sodium reabsorption. We can get to the hypothalamus. And we'll increase thirst and increase vasopressin. So you can see all these things. Once we've sensed that we have a decrease in blood pressure, either by a decreased glomerular flow rate or at the baroreceptor, or there is the direct effect as well, where a decrease in blood pressure just supersedes the pathway and directly stimulates renin production from there. Once we get angiotensin 2, like I said, we vasoconstrict. That's going to constrict the arterioles, and that's going to increase total peripheral resistance, which will increase blood pressure. We're going to increase the aldosterone, which is a hormone that we'll talk about next, and it will increase the number of new channels in the principal cells of the distal nephron, and it's going to reabsorb sodium and help you keep water. So the more water we have, or more water we have into the bloodstream, the more volume, which will increase our blood pressure too. So once we get to the hypothalamus says our blood pressure is low or our blood volume is low. Angiotensin 2 is stimulating us, so we need to increase the thirst mechanism so that we can increase our blood volume. And then we'll increase our vasopressin to basically keep our blood volume up as well. The other interesting thing is we talked about angiotensin converting enzyme inhibitors, ACE inhibitors. Well, there is some side effects with those, because since the ACE inhibitor is working mainly in the lungs, you can get a cytokine reaction to it. So the cytokines is an immune response, and it produces a chemical called bradykinins. And the bradykinins build up in your lungs after a while, and you will have a dry, hacking cough. I personally suffer from hypertension, essential hypertension. It runs in my family. Essential hypertension is hypertension with no known cause, which affects approximately 33% of the population. First line of defense is usually an ACE inhibitor. I had no trouble at low dosage, but once I got to dosage that was actually therapeutic to me, I started getting this dry, hacking cough. It went on all the time. It didn't stop until I ended up taking a new drug. And the drug I take now is an ARB inhibitor, which is, it's a called an angiotensin 2 receptor blocker. So around here, we have angiotensin 2 receptors. And the drug, the ARB, will come in here and bind up the receptor before angiotensin 2 can actually get in there. Therefore, it will prevent all of these effects. So it'll prevent the vasoconstriction, sodium reabsorption, aldosterone release. It'll quiet down the thirst and the vasopressin. So 2 different drugs to manage blood pressure. The ACE inhibitor works in the lungs. ARB blocker works at the angiotensin 2 receptor site and ends up inhibiting angiotensin 2 from actually binding with the receptor. That's all well and good, but we need to look at a little bit more. We've gotten now to the point where we've sensed that there's a decrease in blood pressure. The kidneys have sensed it via a decrease in glomerular flow rate. They've decreased the sodium, the salt transport across the macula densa to help conserve water. The granular cells have been stimulated. They produce renin. Baroreceptors have noticed that there is a decrease in blood volume. They are firing. They'll increase the sympathetic activity, which will stimulate the granular cells once again and produce renin. Since the liver is consistently or constantly producing angiotensin, renin will then bind with, or angiotensin will then bind with the enzyme renin to produce angiotensin 1. In the lungs, angiotensin converting enzyme will bind up angiotensin 1 and convert it into angiotensin 2, which will be in the bloodstream, increase the angiotensin 2 concentration of bloodstream, which will, at the end of it all, vasoconstrict, increase aldosterone, increase sodium reabsorption, and then increase thirst and vasopressin in the hypothalamus. So that's neat, but we did remember talked about aldosterone in chapter 11. So I wanted to go a little bit deeper on that and talk a little bit more about what happens to the aldosterone. So if we remember, aldosterone, or aldosterone, however you want to call it, if we increase concentration, I'm just going to put aldo, we're going to end up with a net increase in sodium reabsorption, an increase in water reabsorption, as well as an increase in potassium secretion. And just for your reference, this is all happening within, we have this loop of Henle. So as things are filtered, different ions are filtered in and out. There's different ion channels back and forth. If we hadn't had weird COVID-19, we'd be talking about how blood would flow in, different things would flow out, come in, stuff like that, the different ions. But we're not going to do that. But we are getting the sodium reabsorption because in order to increase our blood volume, we want to put as much sodium into the blood as possible, because wherever sodium goes, water follows. But the problem is that we're now messing with membranes because we're putting more sodium into the system than needs to be. So we have to balance out the electrolytes. So we secrete potassium. So we get a bit of potassium loss. And this is one of the other side effects of hypertension drugs, is that in the process of tinkering with your endocrine system and your aldosterone and your renin and all the things like that, you end up increasing your potassium secretion so that at the end of the day, you could have low potassium. Low potassium is going to end up messing with your ability to contract muscles. So every 6 months, you go get your potassium tested to make sure that your hypertension drug isn't killing you instead of helping you. So at the end, these are the main functions of aldosterone. Some of the facts about it: it's produced in the adrenal cortex. So now we're back to the kidney again. We have given a mechanism that the kidneys produce, which has gone through all the way through, produced another chemical called angiotensin 2, which stimulates a hormone that is produced at the adrenal cortex of the kidney, or I should say of the adrenal gland. So the half-life of aldosterone is approximately 15 minutes, and it's produced on demand. And you can get a squirt of this when we notice that there is a decrease in blood pressure, as well as an increase in plasma concentration of potassium. The target tissues are the principal cells of the kidney, which are the cells that are producing renin. So at the end, they're going to increase sodium reabsorption, so we can increase our blood volume and increase potassium secretion to see electrolytes. Okay, so been a bit redundant on that, but let's look at it from the picture side. Here's our bloodstream, and in our blood, we sense that there is we had a decrease in blood volume or a decrease in blood pressure. So we've gone through the whole mechanism already, and we have molecules of aldosterone floating around the bloodstream. And this is a steroid hormone. So remember, a steroid hormone can pass through membranes, cell membranes pretty easily. So we have this cell here that lives in the renal collecting duct of the kidney, and this is a principal cell. This principal cell right here. So the principal cell has inside of it has these little triangles like that. And these are aldosterone receptors. And like all good cells, we have a nucleus with some DNA. So when the body has sensed that there's an increase or a decrease of blood pressure and aldosterone's been released, we start seeing this flow through our body, and we've got a 15-minute window, and aldosterone enters into the principal cell. And it's going to end up binding with one of the angiotensin or aldosterone receptors here. Once it binds with the receptor, it then can enter the nucleus. Once it enters the nucleus, it's telling the cell basically, hey, we need to turn on the DNA. We need to get new pumps. We need to get new ion channels. We need to get, essentially, we need to get sodium out of the lumen of the distal nephron and into the bloodstream to increase our blood pressure. So that makes some messenger RNA. That in turn goes to a ribosome, does all the manufacturing process, and we make some new pumps and new ion channels. And what we end up doing then is right now, remember we had our sodium-potassium pump. We had potassium coming in, sodium out. So now we need to secrete potassium. Remember, one of the main things that aldosterone was potassium secretion. So remember, we need to make new pumps, new ion channels. So one of the pumps and ion channels is a potassium specific, and this potassium is going to now be excreted or secreted into the lumen of the distal nephron. So potassium's been secreted at this point. But we have a high concentration of sodium here. And in order to get the sodium, we need to get sodium into the bloodstream because sodium is going to be surrounded by water, and that's going to increase our blood volume. So what we need to do then is the sodium here. We need to increase that. So we bring it in from here. We've made a new ion channel, a new sodium channel, and we get it to come in here. Okay, so we've now increased the sodium concentration, and then our sodium-potassium pump can pump sodium out, and then we can increase the sodium concentration in our bloodstream, which will bring more water around it. And the more water we have, the more volume. And then an increase in volume is going to translate to an increase in blood pressure at that point. So that's basically the function of aldosterone. But this brings us to another hormone that the heart produces. What's really kind of interesting is bodybuilders, and we'll close with this. So now bodybuilders, they want to take advantage of the opposite of this, because bodybuilders, they need to put on a lot of muscle, and they eat a lot of things like that. But when they go to their competition, they need to look as lean as possible, and that means dehydration. They need to get as much of the fluid, this water, out of their system, decrease their volume so that all the fluid that they have is going into their blood. It's being pulled out of the interstitial so that their system looks very dehydrated. All their skin looks very taut, and you can see all the muscles very well. So the heart makes a hormone called ANP, atrial natric peptide hormone. It's a peptide hormone. It has a short half-life, but what it ends up doing is it decreases the sodium concentration in your bloodstream. So if we decrease the sodium concentration, all the water is going to, that surrounding it is going to leave too. So that's very effective way of kind of getting some of our body dehydrated. So the bodybuilder is going to do something really interesting. So about a week out from their competition, they're going to ingest as much salty foods as they possibly can. They're just going to salt it all up. Salt, 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 salt. And the body's going to sense that as basically a chronic increased concentration of sodium. So if our sodium is above the threshold of homeostasis, remember we have a negative feedback loop. So an increased sodium level is going to trigger ANP, atrial natric peptide hormone from the heart, to be produced in order to combat this increase in volume. Because there is nothing wrong with the bodybuilder's blood pressure, but they've eaten so much salt that they've retained water at this point, and that the sodium is being sensed by the receptors in the heart as way too much sodium, and this hormone is now produced, ANP. ANP then, essentially, the end result of it is to cause sodium excretion. So instead of it going this way, sodium is going to be in the cells and and sent into the collecting ducts and being concentrated as urine. The end result of all that is that as the heart is producing ANP and increased concentrations of the hormone ANP are causing sodium to be excreted via urine, it is basically just trying to get sodium levels back to homeostasis. So since the bodybuilder's diet is chronically high in sodium at this point, ANP is continuously being produced, sodium is continuously being excreted at a high level. So the night before the competition, the bodybuilder does something pretty strange. Well, I should say probably a day before the competition. They completely stop eating anything with salt in it. Absolutely no sodium into the system. So what is happening at that point is ANP is still being produced by the heart at a very high level. So you have increased concentrations of ANP. And what it's doing is it's floating through the bloodstream. And since there's no more new salt being added to the system, it's removing all the sodium that's in the bloodstream. And it's taking with it all that water to be excreted as well. So remember, if we have sodium into the system, it's being surrounded by water and it's increasing the volume, the bodybuilder is unnaturally now using the heart hormone ANP, increased ANP concentration while eliminating sodium, new sodium into the system. So ANP is eliminating all the sodium it can in the bloodstream and excreting sodium as well as the water that's associated with the sodium with it. This causes a dehydration state. So now that there's no sodium, or not no sodium, lower sodium and less blood volume, water from the interstitial and the extracellular fluid is going to diffuse into the bloodstream in order to bring volume up. And at that point, that is essentially pulling water from the cells, the skin cells, the cells that are around surrounding the muscle, and dehydrating it and allowing that bodybuilder to see all the definition of the muscles by pulling the water away from them. And that's how that works. So no test on any of this stuff, but I wanted to give you an idea of what walking through a mechanism would be on your final exam paper. So I took this one from Being Sensed. We got the history, a little bit of the history of the system, what happened, who discovered it, why they did it, how they did it, to the actual pathways, what organ systems were involved in it, what was the target systems, and then I took one of the hormones that was produced at the end, and I took that to the mechanism of that. So you don't have to go nearly as deep as I did, but pick something along in there and detail it a little bit and get into the mechanism on that. Cool. Was that helpful? Let me stop sharing that and get back to me. Okay. So was that helpful at all? Useful? Interesting? Feedback? since it's our last class today, I figured we'd do something a little bit different. And if it failed, it failed, and nobody would be the wiser. But I think it worked out pretty well today. What did you guys think of the kidney dissection? Couldn't have done that in class, could we? Yeah, no kidding. Me too. That's really all I have for you. If we, like I said, if we were in class today, we were all there. I would have bought some Krispy Kreme donuts, some fat pills for us, and we could have talked all about the mechanisms of digesting a donut, taking that glucose all the way down and destroying it into 2-3 carbon parts called pyruvates, sending that through the citric acid cycle, making some substrates, sending those substrates over the electron transport cycle, and then producing a whole bunch of ATP. But since we didn't do that, hopefully this sufficed. So that's all I have for today. I'll hang out for a few more minutes if you guys have any questions or feedback. I would ask you, if you do have feedback that you liked the class, you liked we kind of pivoted, don't be afraid to tell the department chairs or the powers that be that you got something from it, you liked it, that we did something good. They only hear the bad stuff. So if you are feeling very generous or you really liked what we did, feel free to tell them. Like I said, they only hear the bad stuff. They love to hear what's going right, just so that. So I'll leave it at this. If you need me in the next few days to a week for your final exam paper, just send me an email and we can do one of these video chats together, and I can help you answer any of your questions that you have. If you don't want to do that, you can email me. And but I guess that the long story short is I want you to know that I'm available and I want to help you with your projects. I want you to get the grade that you want. Okay. I want to be your asset for that. So don't be afraid to reach out. I love this stuff, as you guys know. So Vanessa, I'll tell you what, here's how I'm going to work it. I was going to put up an announcement in the next day or so, but I'm going to go through the grade book more than likely tomorrow or Saturday morning, and I was going to post the final amount of points that we have that will be that you'll be dividing by, so that you can calculate everything. Oh, Chloe, good question. Yeah, I'll put an assignment folder for that. Didn't think of that. Thank you. If you guys do think of anything else, just let me know. Hopefully you know I've been a little bit flexible, so I don't I don't anticipate that changing at any time soon. Okay, if there's nothing else, I'm going to wish you an awesome semester, and glad I got to know you guys and that our paths crossed. And hopefully you learned a little bit about physiology and some things that you'll be able to take on and use the rest of your life. It's been a weird semester, and thank you so much for hanging in there. I think we made as much lemonade out of lemons as possibly could, so hopefully you agree with that. And with that, if there's nothing else, I'm going to close down and wish you guys a great summer and great things in your life. And to the graduating seniors, I'm sorry it didn't work out and your graduation isn't there the way it was. I feel for you. That's a big part of the college experience. But hang in there. You'll soon get a paycheck from a job, and that will make up for it, okay? Talk to you soon. Don't be strangers. If you need anything, please reach out. It is so boring at my house being by myself, and it is fun to talk physiology.